Hi everyone, this is Renee, and this is supposed to be the last session of the almost 1 hour introduction of our department. And our team work on respiratory infection, especially for virus infection. And we start from the epidemiology of respiratory virus in children. So we know how the pattern look like. After that, when we spot some very specific patterns, then we would like to answer multiple questions in terms of the mechanism, this disease mechanism, susceptibility of the host. So we work in wet lab and develop different methods to study the interaction between viruses and the host, and specifically with pediatric cells that I'm going to mention in the later slide. Thirdly, as Jenny just told you, we got little tools. It's a nasal strip that can do longitudinal, quite a frequent sample collection from the nasal area that we can profile different mediators, antibodies, virus dynamic over time with very little pain. So it's non-invasive and can be applied to different projects. And the same things, we submit that to the ITF, which is the innovation grant. And then we are going to apply our invention in the community. So I'm going to walk you through from epidemiology, wet lab, bedside, and then to community. So if you work with me in the team, then you might work in either the Prince of Wales Hospital or the Hong Kong Children's Hospital as introduced by Tom earlier. And we went out to school to collect samples from kids. We do wet lab in the middle, as you see, and the lab in the Hong Kong Children's Hospital is quite new and furnished with all the equipment as Tom mentioned to you earlier. So let me talk about the epidemiology. So this figure is showing you from 2014 to 2020, all the changes of viruses a long time. So these gray lines and black lines shows you the influenza virus epidemiology. As you always heard about, hey, it's time for vaccination. In the past is for influenza and now it's for SARS-CoV-2. You can see that the reason why people ask you to do vaccination is because of the biannual peak that you observe from time to time. But usually people ignore the other things. What is that? That is the blue lines, which represent the enterovirus, rhinovirus that always appear along the year. So it is not only affect the general public, but more importantly, it always appears in pediatric patients. That's why in our department, we would like to study more about the rhinovirus, enterovirus. And this figure shows you the data from 2015 to 2017. How this blue bars, which is representing enterovirus, rhinovirus, is representing in different age group in pediatrics when compared to the very famous RSV in brown, and the gray color, which is influenza and the other viruses. You can see very clearly that the blue bars actually severely affecting those below 1 year old, 1 to 2 years old, and 3 to 6 years old. So we really want to tease out what is the fundamental problems of these subjects and how's the interaction of all these subjects' cells with the viruses. So we develop different cells model. And within this enterovirus, rhinovirus group, apart from the interaction between viruses and the host, it is also interesting to look into the molecular epidemiology. So what I show you this half moon cake, so 42% of them is rhinovirus A, and 7% of them is rhinovirus B, which are the classical rhinoviruses that are affecting half of the population, while the other half, which is orange taste here, is rhinovirus C, and this rhinovirus C is actually contributing to multiple problems. For example, it is suggested to cause a more severe respiratory infection and a higher chance to have association with severe asthma exacerbation. So our group is focusing in RVC and look into that. And sorry to jump with this slide because recently we tidy up the overall 2015 to 2020 data about the virus interference between the influenza and the rhinovirus. And from this figure, you can see that the blue lines are representing influenza, while the red lines are representing the rhinovirus. They are having a occurrence that if the influenza peak early, then the next peak will be the enterovirus rhinoviruses. And they seem to interfere each other. If you have a higher peak this time, then the next flu peak will be lower. So we observe different, we test with different statistical models, and then we also test the same things in vitro. So we have different models doing rhinovirus infection, influenza virus infection, and test how these things interfering each other. So apart from doing that in the population levels, we also do it in the individual levels and also in cellular levels. So how can we study the viruses in vitro? We can do that in small animals. And these are the neonatal mice, which is just 5 days old, and it represents the pediatric airway. And at the same time, from our patients and donors, then we have obtained different kind of epithelium, which is the outer barrier which interacts with virus all the time. So we study the conjunctival epithelium, nasopharyngeal epithelium, bronchial epithelium, and alveolar epithelium, which is from the top to the lower lung. So it represents different regions of infectivity using this model. And with this ex vivo organ culture, we can do multiple temperature, multiple viruses, and test multiple drugs in one shot. And in vitro cell model, so we obtain those organ culture, but at the same time, utilizing the same biological materials, we can isolate single cell type. We develop alveolar macrophage, peripheral blood macrophage, neutrophil, all these circulating cells model to test on the inflammation responses. And at the same time, with the upper to the lower tract epithelium, we test about the interaction of viruses and the host. This picture shows you the nasal epithelial cells that regrow from one single cells, and then we derive them into pseudostratified layer, which means that it contains the Wilbur cells at the top, which are the mucus secreting cells, the ciliated cells as shown in green color, which are the cells that contain cilia that moving along and help you to sweep your dirt from the airway. And at last, you can see the club cells, which are the progenitor cells of the ciliated cells and Wilbur cells. So in general, we collect samples from subjects, we do isolation, we culture them, differentiate them into the pseudostratified layer as you see just now. But at the same time, we also do a little bit one step further, that we culture them in 3D expansion. We expand them in 3D and do it in matrix gel. And at the end of the day, we have the airway organoid done. So what is so special about this organoid? And in this movie, you can see that it's got organized structure, that you have cilia, you got mucus, and then they are functioning in a regular way. So with these very physiological models, you can study whatever kind of respiratory insults or test along the different drugs as long as they are there. And then that can already be a very simple project as a start. And we are not happy to get stopped at that point. So what you see is that all the cilia are facing inside, which is if you imagine you add virus on top of that, then the virus might not touching the ciliated sides, which is not physiological enough. So we do an apical out modification. That means that all the cilia, after these apical manipulations, the cilia are now facing outside. So when you do every kind of insult, then the apical cells are now touching what you want them to expose to, just like you inhale the virus, inhale the bacteria, or inhale your drugs. And we can also do high quality imaging that we even spot individual proteins such as this CDHL3 in red color, which is the receptor for rhinovirus C, as I introduced you in the very early slide. And one of the students is currently working on the cilia beating motions and how it affects by different kind of viruses. I would like to show you that in our culture, we can see a very synchronized cilia movement without any infection. But after infection, you will see that it is dysregulated and the speed are no longer the same, and the movement and the magnitude are no longer the same. So we got different kind of method ready in the lab for you to test on different respiratory insults. And apart from all the imaging, videotaping, indeed we publish paper and have physiological endpoints to assess the usefulness of drugs when some insults are going to create acute lung injury. So we got this transwell system that we have the epithelial cells here, and we put different kind of fluorescent protein there, and we can measure things like the transportation of different size of protein across the epithelium, and also the fluid clearance of the on the epithelium, which are representing the alveolar fluid clearance and also the protein permeability, which are the very critical physiological endpoints that people use to measure. So in this particular paper, we are using mesenchymal stromal cells to help reversing the severe outcomes caused by the influenza virus infection. And lastly, I would like to introduce you the little tools, nasal strip to collect the mucosal fluid. So this is a very short, kind, and soft paper material that can insert into children's nostril, and it's optimized for different uses, including virus detection, antibody detection, inflammatory cytokine, and hormones detection. And we also preliminarily test that it can have enough biomass for microbiota testing as well. So this is a non-invasive method that we can use on different subjects with different diseases, and it is stable and can tolerate long-term room temperature storage. So in case you're going to have community study or sending all these little tools to your patient and ask them to mail it back, it is now possible. So it opens up many opportunity in profiling something which is dynamic and we don't know in the past. And I think this would be a good opportunity to translate all this data into a better understanding of the pathogenesis of different respiratory diseases. And these are just some examples showing you how we use this nasal strip to do viral detection during SARS-CoV-2 period. And it is comparable to all the nasopharyngeal swab that people did, and also is somehow could be better than the deep throat saliva, in particularly when children are not able to provide a very good saliva sample. At the same time, with the same tools, we also do the characterizations of the mucosal antibody. So why is this important? If people always study serological antibody, thinking that that could represent how much it protects the host, but indeed that is responsible for the control of severity. So mucosal antibody is actually the key to limit the infection at the upper airway. So with this nasal strip, we are able to study the changes of the mucosal antibody a long time and along the disease course. At the same time, another study that we have already submitted for review is the how the mucosal antibody changes after vaccination. So we investigate 2 different vaccines which are available in Hong Kong. One is CoronaVac, the other one is the BioNTech that you are very familiar with, and we see the changes a long time and compare the 2 different vaccine efficacy. So at last, I'm going to show you how to make good use of this in a translational manner. So when we do antigen tests, you can buy it commercially with this little rapid test kit, and they just test for the viral antigen. And now with the data we found, actually with the mucosal antibody that's secreted after infection or secreted after vaccination, we can, apart from doing the standard antigen test, the same material can be used to test mucosal antibody, which is one sample and two answer for the presence of antigen and antibody. And these are the project team members that our team used to work together with, and they provide professional lab consultation and also recruit patients and the subjects for us. And these are the team members of my team, and 3 of them are students, 2 of them are research associates who has their PhD degree done several years ago. And there's many more undergraduate interns locally and internationally that comes and go, and we interact happily together. Thank you very much for your attention, and that's all for my talk.